To introduce Leo Bonilla. Is that correct? Is that how you say it? Leo Bonilla. Okay, great. So he is a professor of neurology and a clinical scientist at Emory. So he's right down the street from the Trend Center here, and he's a clinical neurophysiologist and epileptologist, and he has also been trained in computational neuroscience and neuroimaging as well. So he's very focused on the brain and on epilepsy. And today he's going to talk to us about focal epilepsy and interfacing that with artificial intelligence. And I think a little bit more of an emphasis on sort of motivating why we want to study epilepsy with these kinds of tools, and very much looking forward to your talk. So thanks again for joining us, and I'll pass the baton over to you, Leo.
Thank you very much, Vince. Thank you, Anise, as well, and Armin for the introduction. It's a pleasure to be here. So I am going to talk about focal epilepsy and the motivations for using artificial intelligence in the context of neuroimaging for diagnosis of epilepsy. And just as starting with disclosures, I don't have any conflicts of interest associated with this talk. None of the products or the tools that I'll talk about today are commercial. I don't make any money associated with any of the methods that I'm going to be describing. Of course, many of the things that I'll talk about today entail MR scanners, but I don't really have any conflict of interest associated with those. I do research on two topics, language and aphasia being one of them, and epilepsy being the other one. Obviously, in the context of the talk today, I'm going to focus on epilepsy only, but the research support that I receive is mostly from the NIH, and it's listed here. And again, as I said, it's related to epilepsy, some, and language and aphasia in the other part. So the outline for this talk is I'm going to emphasize for maybe a quarter of the talk, or a little bit more, the significance of neuroimaging in the diagnosis of epilepsy. And in particular, I'm going to focus on one form of epilepsy called temporal lobe epilepsy, which is by far the most common form of epilepsy in adults. And it's also the most common form of medication refractory epilepsy. So that means epilepsy that does not become well controlled with medications. And then I'm going to talk a little bit about quantitative neuroimaging literature, so research in neuroimaging in temporal lobe epilepsy, so beyond what's done clinically. And that would be used as a backbone for the motivation for using artificial intelligence, which is a novel thing in the context of neuroimaging research in epilepsy, and some innovations, recent findings from our group, also in collaboration with Anise, Vince, and the group at Trends, and then eventually where to go next. So in summary, I'm going to spend a good amount of time, particularly in the beginning of the talk, talking about the clinical importance of the problem. So why is it that we need better neuroimaging, and why it is that we need artificial intelligence in neuroimaging? What is the clinical problem we're trying to solve, and what's the impact of this research? So there's a neurological disease called epilepsy. Epilepsy is defined by recurrent and unprovoked seizures. So seizures, by definition, are an abnormal electrical rhythmic activity in the brain. Somebody can have a seizure as a consequence of having a head trauma, as a consequence of having a metabolic derangement. So, for example, too little sodium in the blood, too much sodium, etc. But there's a disease in which seizures are not provoked. So they're not caused by head trauma, they're not caused by a metabolic derangement or anything like that. And so they're spontaneous and they're recurrent. So that's the definition of epilepsy. And within epilepsy, there are essentially two main types. One is what's called genetic generalized epilepsy, which I won't talk about it today. And that's a genetically defined form of epilepsy. And it's relatively less common, albeit somewhat common, but less common. And the other type of epilepsy, which is the one I'm going to spend most of my time talking today, is what we call focal epilepsy. And that is defined by seizures, again, the spontaneous seizures coming from one part of the brain. Focal epilepsy is the most common form of epilepsy, and within focal epilepsies, medial temporal lobe epilepsy is the most common cause, right? So again, just to recap again, if you take all forms of epilepsy, you have focal epilepsy and generalized epilepsies. And if you take focal epilepsy, you have medial temporal lobe epilepsy being the most common type. By definition, medial temporal lobe epilepsy is associated with seizures that come from the medial part of the temporal lobe. Most commonly, that's from the hippocampus or coming from within the hippocampus, but it can also mean the areas close to the hippocampus, such as the fusiform gyrus or the entorhinal cortex or the perirhinal cortex, and less commonly from basal temporal region. So as you can see here, the uncus, which is the medial surface of the medial part of the temporal lobe, is painted in blue here, and this is the entorhinal cortex. So more specifically, if you look here at a cut of the brain, this is where the hippocampus is. This is the entorhinal cortex. This is the perirhinal cortex. And seizures coming from this region, broadly stated, are coming from the medial part of the temporal lobe, and some folks with medial temporal lobe epilepsy have seizures coming from those regions. Medial temporal lobe epilepsy is the most common form of epilepsy. It affects approximately 1 million people in the United States, and it's estimated that around 50% of all patients with epilepsy have medial temporal lobe epilepsy. So if you know 3 people with temporal lobe epilepsy, 3 adults with epilepsy, chances are that at least one has medial temporal lobe epilepsy. And again, it's a very common form of epilepsy. The typical clinical presentation associated with medial temporal lobe epilepsy is composed by 3 different types of seizure semiologies. And semiologies is the term that we use for the clinical manifestations associated with seizure. So folks with temporal lobe epilepsy, they may have what's called a simple focal seizure, and that may or may not progress to a complex focal seizure, which may or may not progress to a generalized tonic-clonic seizure. So what are these things? A simple focal seizure is what we call an aura. In temporal lobe epilepsy, it's typically of this limbic characteristic, and it's often associated with epigastric discomfort. So that means, it's upset stomach. Sometimes associated with autonomic features, so that can be salivation, pilorection, etc. And there can always more commonly be perceptual manifestations such as anxiety, déjà vu, which is this feeling of odd familiarity with the context in the present that shouldn't be that familiar, or jamais vu, which is the opposite. And sometimes there can be hallucinations, so the abnormal perception in terms of taste and olfaction, usually unpleasant. Complex focal seizure is the second one, and by far that's the most common manifestation of medial temporal lobe epilepsy, and that's associated with loss of awareness or loss of consciousness. Consciousness is obviously a very complex topic, but in the context of neurology, in the context of epilepsy, we define consciousness as being lack of responsiveness and awareness. So in other words, a person not remember what was going on during a given period of time and not being able to respond. So usually that's associated with a motor arrest, with a blank spell, right? If a person is just not responding and you try to speak with that person, they are just a blank arrest, a blank spell or motor arrest. And there can be some verbal or motor responsiveness can be sometimes repetitive. And sometimes that can be wrongly interpreted as being preserved awareness, but it's usually automatic and it's usually not conscious. Sometimes there can be a ipsilateral manual automatism. So that means in the same side of seizure onset, that there can be automatisms like hand picking, etc. And oftentimes there's also lip smacking. The diagnosis of temporal lobe epilepsy is predicated on the observation of a seizure coming from the temporal region on EEG. So to definitively diagnose somebody with temporal lobe epilepsy, you need to get an EEG during the time of the seizure and identify that rhythmic abnormal pattern during the seizure at time locked with the event. And this is how it looks like. So this is an EEG, this is scalp EEG. This is an AP bipolar montage. So it means you have electrodes that are placed around the scalp, and one electrode is compared to the next. So this is the temporal chain here. So all these electrodes are in the temporal chain on the left-hand side. This is the perisagittal chain, so close to the middle on the left-hand side, central, perisagittal right, temporal right. So you can see left to right, going from the top of the page to the bottom part of the page. And what we see here is at a given point, probably around where this red arrow, the red line is, you see a rhythmic theta pattern appearing on the left side of the brain. You see right over here. That rhythmic theta pattern that evolves is very characteristic for a temporal lobe seizure, and this is a electrographic scalp evidence of a rhythmic pattern that is compatible with a seizure with origin on the left temporal chain. So again, this This is a seizure documented by EEG, and you need this for the diagnosis of epilepsy. The most common cause of medial temporal lobe epilepsy, it's what's called hippocampal sclerosis. Hippocampal sclerosis is a histological abnormality. So again, you can see signs associated with hippocampal sclerosis on imaging, but by definition, hippocampal sclerosis is a histological abnormality. So you need to look at, a slice in a microscope to diagnose that. And it is defined by cell loss and gliosis involving some subregions of the hippocampus, particularly CA1, CA3, CA4, and the dentate gyrus. Right, so there's usually relative sparing of CA2, but that's how it is. Usually, hippocampal sclerosis is pronounced enough that you can see in macroscopic brain slices. So here is a macroscopic histological slice. And you can see here a hippocampus on the left-hand side, a hippocampus on the right-hand side. And assuming that we're looking at the brain from behind, right? And you can see here that on this side, there is cell loss and gliosis, and it's quite noticeable how atrophied this structure is in comparison with the hippocampus on the other side, right? And with hippocampal formation and the surrounding structures on the other side. It just so happens that this pattern is pronounced enough that it can also be seen with clinical MRI. This is a fairly old MRI, clinical MRI, and you can see here quite clearly that on a T1 weighted scan in the hippocampal region, there is atrophy of the hippocampus. There's also signal loss within the internal part of the hippocampus. This is commonly referred to as loss of internal architecture. And that essentially means hypointensity of the T1 signal within the hippocampus, right? So it's dark. And that's that. Oftentimes, this is also associated with increased signal on T2 weighted scan. So you can see here the same scan, and this is the T2 counterpart, just showing the part of the temporal lobe. And you can see quite clearly there's a lot of increment in T2 signal in the same region. The combination of increased signal on T2 scans, atrophy, and loss of internal architecture are the hallmarks of the definition of signs associated with hippocampal sclerosis on MRI scan, but you don't need all of them. You need just a few, you need one or the other to identify signs associated with, again, hippocampal sclerosis, which is a histological abnormality. Commonly, it's believed that atrophy of the structure is more associated with cell loss, whereas increased T2 signals are more associated with gliosis. But again, both things are compatible with the hippocampal sclerosis. It's very important to know about temporal lobe epilepsy because the response to medication in temporal lobe epilepsy is suboptimal. So again, not only it's the most common form of epilepsy in adults, but it's also one of the most common forms of epilepsies that do not respond to medication. So there have been a few classic studies in the field of neurology and epilepsy evaluating this pattern. This is one of them. This is a study from 2000, if I'm not mistaken, 2000 or 2021. And it looked at the likelihood that patients with epilepsy, then the vast majority in this study here had temporal lobe epilepsy, responded to seizure medications. And you can see 47% of patients responded to the first line of anti-seizure medications. Of those who did not respond, they went on to take the individuals who did not respond went on to take a second medication. Of the entire number of individuals, an additional 13% responded. So it's not 13% of the ones who did not respond, it's 13% of the entire group. And then those who did not respond, who went on to try another medication or more combinations, just 4% did not respond. So this is the same idea just shown by at a different paper a few years later. And you can see here the percentage of individuals that respond to the first anti-epileptic medications, is around 50% for the first medication, and it drops, it goes up to close to 60% for the second one, and then it plateaus after that. So there are 2 points here. One is that there's a lot of individuals who did not respond to medication, right? And also those who don't respond to the first line of medication or the second line are very unlikely to respond to subsequent trials of medication. If they fail the first one and the second one, it's unlikely that a third, fourth, fifth, sixth, seventh medication is going to work. Good news is, for individuals with temporal lobe epilepsy who did not respond to seizure medication, as we saw, that number is around probably 30-40%, surgery can be extremely effective in terms of yielding seizure freedom. surgery can be curative to epilepsy. And this is a very, very important point. Surgery for temporal lobe epilepsy has been traditionally performed by removing a part of the brain where we expect seizures to come from. This was originally defined in the 50s as a anterior temporal lobectomy, which is a resection of the interior part of the temporal lobe, trying to maximize the resection of the medial part of the temporal lobe, but also with resection of some structures around or in the lateral part of the temporal lobe. You may ask, well, if the seizures come mostly from the medial part, why do you take some of the stuff outside, some of the lateral temporal cortex? That's a good question. That's because the surgery was originally defined like that, and more recently it's been refined to some extent to target mostly medial parts, medial areas of the temporal lobe. There are a few variations to that. There's a surgical amygdala hippocambectomy. The same procedure can be done with laser nowadays. But by and large, that's the idea. The idea is to remove that part of the brain that is generating seizures. The good news is that this works. Whatever variation of surgery that I was talking about before. This was one of the only randomized controlled clinical trials in epilepsy or in temporal lobe epilepsy with regards to treatment. And you can see here that the percentage of individuals without seizures impairing awareness in the surgical group was much better compared to the individuals in the medical group. So in other words, 58% in the surgical group here became seizure-free, whereas only 8% in the medical group became seizure-free. So this is a gigantic effect, right? Patients are significantly more likely to become free of a lifelong disabling seizure with surgery. So surgery can be not only curative, but transformative to many patients. For this reason, a few years after that clinical trial that I mentioned before, the American Academy of Neurology issued a practice parameter statement. So in other words, a guideline for clinicians saying that temporal lobe epilepsy should be treated with surgery if medications have not worked. And more specifically, what this practice parameter said was, if somebody has not achieved seizure freedom with 2 first-line seizure medications, they should be referred for surgical evaluation within 1 year. So in other words, try one medication, try another one. If that has gone for a year and somebody has not become seizure free, then they should be referred for surgery, because surgery is likely to yield significant benefit, and keep trying other medications is not going to work. There have been a lot of emphasis or additional points to corroborate the importance of surgery. This is an interesting paper. If somebody's interested in looking at this is a paper by Sam Veeb, who's one of the authors of the early studies about epidemiology of epilepsy and also surgical treatment. And he mentioned that surgery for temporal lobe epilepsy is probably one of the most efficient treatments in modern medicine. It's really important to consider it, and that should be strongly thought about, and patients should not be trying multiple medications if surgery can be done. So far, so good, right? I think one of the things that I want to emphasize is that temporal lobe epilepsy is a condition that is very prevalent. It can be associated with significant improvement if the diagnosis is done correctly. But importantly, neuroimaging plays a significant role in the diagnosis. As I said, the diagnosis of temporal lobe epilepsy is predicated on the identification of seizures on EEG, but since the vast majority of patients with temporal lobe epilepsy have hippocampal sclerosis, the identification of signs associated with hippocampal sclerosis on imaging is paramount, because it provides further assurance about not only the type of seizures, but the etiology of seizures, right? So where the seizures are coming from. As I said before, the vast majority of patients with hippocampal sclerosis will have atrophy or signs on MRI that are associated with hippocampal sclerosis, and atrophy on T1 weighted images and increased signal on T2. So you can see here very clearly another example of the same thing. However, that is not what's commonly seen all the time. So let me go back real quick. It's not difficult to identify signs associated with hippocampal sclerosis when these is the pattern that we see. Unfortunately, many individuals have much fainter signs, much more subtle signs on imaging. As you can see here, there is not, this is again a G1 weighted scan, right? There's not a significant pattern of atrophy on one hippocampus versus the other. Maybe there's a little bit of malrotation in the left temporal region here. So the angular bundle, which is this area here, is a little bit more verticalized compared to the other side. But still, there's not a lot of atrophy on this hippocampus compared to the other. Yet this person had temporal lobe epilepsy and became seizure-free after surgery. So you could still have temporal lobe epilepsy. You can still have normal imaging based on visual qualitative assessment and have similar good outcomes with surgery. Let me give you another example. This is another person with temporal lobe epilepsy who also became seizure-free. I think this person was after laser ablation. And as you can see, there is not a very clear-cut pattern of atrophy on by visual inspection of the medial part of the temporal lobe. So what I want to emphasize here is that even though many patients have a pattern of atrophy like this one, many patients do not. They have images that, upon qualitative assessment, they look normal. They don't look abnormal, again, based on just visual interpretation. This is what we refer to as non-lesional temporal lobe epilepsy. So again, these are patients who don't have a quote-unquote lesion on MRI, and yet they have temporal lobe epilepsy just the same. They can have comparable levels of surgical success, but not being able to make this diagnosis with imaging, as you can imagine, can create some issues with regards to diagnosis because there's uncertainty about that diagnosis, right? Is this really what this person have, etc., how to proceed. So, as I said, non-lesional cases can achieve comparable treatment benefits. There are several references to support that. And it then becomes a little difficult in terms of the radiological assessment of these cases if there's subtle atrophy or if there is bilateral atrophy, right? As you can imagine, if somebody has atrophy involving both sides, it's hard to tell which one is more atrophy compared to the other just by visual inspection alone, which, by the way, is the standard of care in clinical setting. And this can generate uncertainty about diagnosis. And it's well documented that uncertainty about diagnosis usually leads to incertitude about next steps and, should there be intracranial electrode replacement, which is oftentimes done as a next step. But if you don't have a good hypothesis backed up by radiology, sometimes electrode replacement can be less clear or less well defined in terms of the planning. And there can also be skepticism about the possibility of optimal or near optimal outcomes, which, as I mentioned before, can be achieved. So this is a big issue, right? This is an impactful problem. If we can fix or if you can improve the radiological assessment of temporal lobe epilepsy, it would be possible to improve accuracy in terms of diagnosis and get people better and faster to the treatment that they need. So moving on to research now. And by the way, I wanted to mention that the fact that we can visually identify specific patterns also prevents us from having a better idea about potential subtypes of temporal lobe epilepsy, meaning that some individuals may have a little bit more atrophy here, a little bit more atrophy there. But I won't belabor that too much. It's a bit of a finer point from a clinical standpoint, but it's also another limitation related to the same point. So can we identify subtle patterns and can we identify other features beyond the hippocampus that could help us with diagnosis? So again, this is where research comes in. Most of the stuff I'm going to talk about now are not used in the context of clinical, in the clinical setting, but they have been done as far as research to try to understand the pathophysiology of epilepsy, just trying to understand what are the mechanisms associated with disease. So a lot of what I'll talk about now is neuroimaging research. So as many of you are familiar with, one way to measure damage in the brain is by using manual morphometry. This is obviously a very old technique, and many of you may have never had to do that during if you've come to neuroimaging more recently. But older folks like myself, we have, and this is what I did when I was doing my PhD 20 years ago. And the idea here was that you can measure the volume, or you can measure the shape of a given structure, and that is linearly associated with cell loss. So for example, this is a hippocampus of a person without epilepsy. And you can see here again, this was manually delineated in the computer, and then from that manual delineation, you can generate a volume. And this is the computer for somebody, excuse me, this is the hippocampus somebody with temporal lobe epilepsy. So you can see there's a quantifiable difference. Of course, we already know that temporal lobe epilepsy is associated with hippocampal sclerosis. So you can use manual morphometry to increase your detection. But the question is there more damage beyond what we can actually see, with our naked eye? And if you use the same method, again, manual morphometry for other regions within the medial temporal lobe. So this is a protocol that we did many years ago to look at structures in the temporal lobe, in the medial part of the temporal lobe, such as the rhinocortex, perirhinocortex, etc. One thing that became apparent back then was that, yes, indeed, the hippocampus is atrophied in temporal lobe epilepsy as a group, but so is the entorhinocortex and so is the perirhinocortex. So these are Z-scores in patients with epilepsy, obviously in comparison with controls. So these structures are really difficult to eyeball, right? Meaning to identify atrophy, but you can measure them and you can identify that it's indeed there is an extent of damage that goes beyond the hippocampus and involves those regions. It is just when you measure it, you're able to see it. Moving on from manual morphometry, which is obviously labor intensive, radar dependent, etc. Automatic measures or automatic ways to evaluate brain tissue are preferred. So this is, as many of you are familiar with, the way to go about, for example, if you want to do tissue assessment with voxel-based morphometry, right? So first you extract the brain from the skull and then extract specific tissues from the brain. So here in this case is gray matter or white matter and CSF. And if they're all placed in the same stereotaxic space, you can compare voxel by voxel, the concentration or the volume between regions. And this is often done after smoothing spatially to avoid individual variability in terms of foci and gyri positioning. And as you're familiar with, voxel-based morphometry essentially works on a voxel by voxel basis, evaluating the distribution of signal intensity within that voxel between groups. So if you see here temporal lobe epilepsy versus controls, for this particular voxel, this is the distribution. You can do a t-test, for example, in this case, and you see the t-value associated with that particular voxel. Every single voxel that you test gets a t-value. You correct for the number of multiple comparisons, and you get a pattern in the brain that is associated with where there are group differences. So doing that with temporal lobe epilepsy, it was this very interesting, again, this is I think it was a revealing study back then, that if you do that with temporal lobe epilepsy, there is atrophy in the hippocampus, but there's also atrophy in the limbic system. And this is quite well defined. This has been replicated numerous times since then. And you can see atrophy that involves the entorhinocortex, involves the basal temporal region, also lateral temporal region, the insula, the thalamus, the anterior cingulate, and so on and so forth, right? So this is very interesting because it provides confirmation that there is an anatomical pattern of atrophy. This here is to show that this atrophy tends to get worse as individuals have had longer years of epilepsy. There is an anatomical logic to this. So nuclei in the thalamus that are more associated with the hippocampus tend to have more atrophy. So this is not a willy-nilly pattern of atrophy, but it's associated with limbic connectivity. So this is the anterior nuclei versus the posterior nuclei, and you can see the anterior nuclei, which are connected to the hippocampus or the limbic system, tend to show a lot more atrophy. And it involves not only gray matter, but as you expect, can also involve white matter. So respectively, orange and blue here in this slice. So in summary, you can identify signs associated with hippocampal sclerosis with visual inspection MRI. This is very similar to the visual inspection about gross histological cuts, and this is very helpful. Yet there is more than what meets the eye with regards to atrophy, and if you already use quantitative methods such as manual morphometry or slightly more sophisticated with tissue volumes such as VBM, voxel-based morphometry, or surface thickness, etc., it's now recognized that there's a limbic pattern of atrophy that is quite well defined, and white matter microstructure also tends to be follow along. You also have a pattern of white matter damage that is associated with damage. So this is with regards to the quantitative neuroimaging literature thus far. So what we know is that there is indeed damage that extends beyond the hippocampus. The abnormalities tend to follow an anatomical pattern within the limbic system. They involve gray and white matter, and they tend to be worse with duration of epilepsy. And I didn't show a picture here, but they also tend to be worse in the context of dominant hemisphere temporal lobe epilepsy. So somebody who's left hemisphere dominant for language, if they have left temporal lobe epilepsy, usually the pattern of damage is a little bit more pronounced than if they had right temporal lobe epilepsy. So even though this is a interesting pattern or a pervasive pattern, it has not been used for diagnosis yet. This has not been leveraged for improving clinical assessment of epilepsy. A couple of things I want to just also talk about in the context of this pattern is that it tends to be quite symmetrical between left and right temporal lobe epilepsy. So this is right temporal lobe epilepsy, right side of the brain, left side of the brain here. So this is a radiological convention, right? So you can see here where the atrophy is. And if you go to left temporal lobe epilepsy, it's almost exactly the same, except for mirrored, right? So this is a very typical pattern that we see. And also importantly, and I think probably this is one of the most important things about this, is the fact that it also tends to be quite prevalent. So what I mean by this is that it's not one or two individuals with temporal lobe epilepsy that will have a lot of limbic atrophy. Most patients with temporal lobe epilepsy will have some degree of limbic atrophy. This is a voxelized area under the curve depiction of patients with epilepsy versus individuals without epilepsy. As you know, the area under the curve is high when you have a significant separation of the distributions between groups, right? So you have a really high AUC, so the area under the curve of a receiver-operated characteristic curve is very high if one group has almost everybody in one group has less than almost everybody in the other group. So in this map here, the voxels that are painted yellow or red, that means that mostly everybody in the temporal lobe epilepsy group had less gray matter compared with individuals that were controls. Again, to show that there is a pretty significant high prevalence of atrophy in temporal lobe epilepsy compared with controls. And as I said before, even though this is there, it's quantifiable, it's pervasive, it's common, has not been leveraged for a diagnosis. So as you can see, I'm setting the stage for can we do that, right? So can we leverage this for diagnosis? The theoretical framework here, therefore, just to summarize everything that we talked about before, is that in individuals with temporal lobe epilepsy, whether there is hippocampal atrophy or not, visually defined on MRI. So in other words, if you eyeball the MRI scan from the clinical ground, if you can or if you cannot see hippocampal atrophy, there is a pattern of limbic atrophy that is present, and yet is very difficult to see just by looking at the scans. So again, this is an individual, for example, here, and this is a diagrammatic representation. This is a diagram, just for illustrative purposes. So you can see if areas of atrophy were represented in red, you can see there could be atrophy on the hippocampus, but also atrophy within the limbic system, or there may not be atrophy in the hippocampus visually defined, but there's also this pattern of limbic atrophy that, as I mentioned before, is quite prevalent and yet not visually identifiable. So can we use artificial intelligence to leverage that, right? As you can see, there are variations across individuals with regards to the pattern, but there is a noticeable pattern. And if we were to do that, how can we go about doing it? What is the best method, right? So to start this question, one of the things that we did was, what if we did not use voxel level data? What if we just use region of interest data, right? Do we really need voxel-wise anatomical specificity? Maybe it's just, just look at the atrophy of the entorhinal cortex as a whole, as a group, and that might be enough to identify, pick up temporal lobe epilepsy versus not. I'm sure you guys are familiar with Enigma. There is Enigma epilepsy. So we look at Enigma epilepsy data here. So there were data points from 16 Enigma sites. There were 336 patients with temporal lobe epilepsy, 187 left, 149 right. And then we had 631 controls. And long story made short, using ROI level data, what we found was that the accuracies were 75% plus or minus 3% with spore vector machine. Again, you use for every individual, you get a vector of values, right? Each one element in that vector is related to the volume in one brain region. And our accuracies were 73% with deep learning. And what we did deep learning here was just a feed forward simple perceptron. I think it had 1 or 2 layers, deep layers, and that was it. So not convolutional neural networks or anything like that. Just again, a feed forward perceptron, and that was it. So ROI level data, 1 vector per individual, really did not yield high accuracies. This was interesting. It would have been great if we had higher accuracies, but it kind of corroborated our hypothesis that you really need that fine anatomical distribution or pattern within the limbic system to be identified to generate the diagnosis. So again, if we were to go back to the previous slide, as you can see, it varies a little bit across individuals. It doesn't really respect, specific anatomical boundaries. But if the brain were a canvas, right, you have a pattern, just like a 7 or an 8 or a 6, right, a handwritten digit, and that pattern needs to be interpreted. So let me go back. Sorry, here. So this is exactly what we then thought about in the context of using artificial intelligence. So again, if you consider the brain being a canvas in standard space, right, or in a comparable pattern, as you're familiar with, this is very similar to the issue or to the topic or the challenge about recognizing handwritten digits on a canvas. You have slight variations in the way the 9 or 8 or 6 is written, and yet there's a similar pattern across different digits, and you could potentially abridge that for a diagnosis. So you can imagine here, these are different 6s, right? Just to give an example, and yet they're both limbic patterns. So I don't need to spend too much time here with this for this group, but as you know, this is a handwritten digit recognition or computer vision is where convolutional neural networks excel. And as you know, convolutional neural networks entail applying filters to images. So, if this were your filter and you convolve that filter over a given image, this is what you get. So this is a filtered image on the right-hand side. Again, I'm sure you guys are way more familiar than I am with this. But again, this is a image filtered by this filter, and convolutional neural network essentially entails applying a large number of filters that are initially randomly defined and eventually fine-tuned so that they can be used for classification. So as you know, a simple architecture is apply a convolutional filter, rectify the output, and apply convolutional layers again until you can pull those to a fully connected layer that can then be used for diagnosis, right? And of course, as you do this, you're going to get an error, and the error comes from the right answer minus the predicted answer, and you can use this to adjust, excuse me, each and every one of those elements in your architecture, which again can be very simple, but it can be also very complex. And the weights in the fully connected layer then define the classification for each one of those, and you can fine tune the hyperparameters in each one of those elements here to again further refine the model, and that's really where the tweaking comes in, for lack of a better term. So in this context, we recently published a first study in terms of temporal lobe epilepsy and convolutional neural networks applied for voxel-wise data, T1 data specifically, for identification, for classification of temporal lobe epilepsy versus controls. So this is data that came from 3 different sites: the Medical University of South Carolina, which was where I used to work until I came to Emory, University of Bonn in Germany, and then we had 202 controls from the same places. There were 95 patients pulled from these institutions, and everybody here had left temporal lobe epilepsies. This was the first study we wanted to look at just one side to avoid issues with hemispheric asymmetry and things like that. We use a very conventional approach with regards to machine learning, which is, you split the data into 80% training and validation. You had tenfold cross-validation within training, and then the highest performing model was the one that was used for testing. And then to test for statistical significance, we did the same thing, except for we shuffled the labels here in the training group. excuse me, and that gave us an idea about what a random group, how a random group would be performing, so that we could compare whether it's statistically significantly better with real data compared to random data. And what we did here was very, very simple in terms of convolutional networks. We had 3 convolutional layers, and we essentially look at 2 different kinds of architecture. Incidentally, what we fed into the convolutional neural network here were gray matter maps in standard space. Again, derived from T1 weighted scans. So isometric T1 weighted scans, all of them with 1 by 1 by 1 millimeter in terms of voxel size, but using I don't think we use Cat12 here. I think we just used SPM normalized segmentation parameters and get the probabilistic gray matter map. And this again, what was fed into it. One thing I should mention is we compared 2 different types of architectures. One was the conventional 1, 2, 3 convolutional layers, but we also use a directed acyclic graph architecture in which we also use the first layer and shunted the weights to the last layer so that to maintain some of the information in the filtered images with regards to smaller areas of the brain. As you know, with multiple layers, you can make your filtered images become coarser. But in the context of trying to identify things that happen in small little bits of the brain, in particularly in the rhinal cortex and the hippocampus, it may make sense to shunt the weights from the first layer all the way to the last one, and this is what the DAG architecture came in. So directly cyclic graph, as you know. So these were the feature weights that we observed. So again, this is multiplied by each one of the images, and this is an average from patients. So this is the weights from the first convolutional layer, second, third, and this is with the DAG layer. So you can see here higher classification importance is shown in red, and interestingly, as we anticipated, both temporal regions were important for classification. What was interesting is that the right temporal lobe also appears to be important. If we were to anthropomorphize a little bit what the computer is doing, I think maybe it's trying to compare one side with the other, or maybe at least identifying that there is atrophy in one compared with the other side. That's a guess, of course, something we can test. And this is when there's no DAG, right? So the coarse or image features, but again, the temporal lobe is where the majority of the weights that are important for classification come in. What we found was this, and I want to take just one quick minute here on the slide, and I'm wrapping up. This is everybody, all temporal lobe epilepsy versus controls. These are the patients with lesional temporal lobe epilepsy, the ones that had visually defined hippocampal atrophy. But I want to focus here on the ones that didn't. So these are patients who had non-lesional temporal lobe epilepsy. So those who in visual evaluation of their MRI scans, a radiologist could not tell there's atrophy or not, right? But these are individuals who became seizure-free with surgery. So these are individuals that we know for a fact had temporal lobe epilepsy coming from that area. So again, my gold standard here, my ground truth is not a human. Right? And I think this is really important to emphasize because we know humans don't do very well in this task. And matching the human here is not a good idea, right? What we want to do is do something that's better than what the human can do so it can help the humans, right? That's really the main motivation for this. And if I look here just at the positive predictive value, right? So how likely it is that somebody was going to be able to identify if somebody had temporal lobe epilepsy or not. Human is blue, and then all the other models are convolutional neural networks. NS and SM are non-smooth and smooth data, so you can feed the gray matter smooth or not smooth. And this is with DAG, right? So you can see DAG did a pretty good job here for a positive predictive value, and you can see in just with regards to the area under the curve, it's much better than humans because, as I said, humans don't do very well with this. So I think this is really, really reassuring. I think this is encouraging, probably better said. So at a glance here, humans had 68% accuracy, convolutional neural networks at 88%. In non-lesional TLE, which humans don't do very well, it's closer chance, but convolutional neural networks still did quite well. One important subsequent study, and this is done with Vince's group, Anise as well as part of this, is to compare with other diseases, right? Because this might just be a non-specific pattern associated with epilepsy. So we compare temporal lobe epilepsy with Alzheimer's disease and controls. Convolutional neural networks, again, two-dimensional convolutional neural networks, I should mention that, did a pretty good job in terms of separating those. And at a glance, the temporal lobe epilepsy patients can be classified accurately within this three-group classification 81% of times. Alzheimer's disease, close to 80%. And lateralization is something that we're observing now that can also be done with pretty good accuracy. So left versus right temporal lobe epilepsy. This is still work in progress. So in conclusion, whole brain network imaging of epilepsy has provided an important framework for a decision support tool that could potentially be used to augment the detection of features associated with temporal lobe epilepsy. As I said, many of these things are not, they're prevalent, there's a theoretical framework, there's a neurobiological reason for existing, there's a neuropathological pattern that is well defined now with neuroimaging research, and yet we don't use this for diagnosis, and we could if we leverage artificial intelligence for this. So there's great translational potential. So next steps, and this is stuff that we've been working with the group here. I'm really pleased and thanked for having the opportunity to work with Vince, Anis, and Rehani on this. Is we can use more sophisticated convolutional neural networks. We can identify subtypes of temporal lobe epilepsy, response to medication. It's also, as you can imagine, a big question, right? And then also apply multimodal imaging. So these are all subsequent steps in this progress. I should emphasize the importance of Enigma for this. Enigma epilepsy certainly provides the numbers of patients that we need for this type of analysis. Epilepsy is not as common as Alzheimer's disease, thankfully. Thankfully for epilepsy, it's not as common disease. So pulling data from multiple sites is really important so we can have better numbers and better train the model. And then I do have a separate study on connectome imaging and temporal lobe epilepsy that also has been partnered with epilepsy that we use data for this particular study. That's a separate study that's called, we nicknamed ESCAPES, Connectome Abnormalities to Predict Epilepsy Surgery. So it's a multi-site study as well that also can potentially, or has been providing data for this study. So these are the folks from the lab at MUSC. Many of them have moved here to Emory. So this is a picture that I need to update. But I want to thank many of the folks that are in this slide who did a lot of the work that I showed here. And that's it. So thank you very much for inviting me. I really appreciate the opportunity.
Thank you, Leo. That was great. So let's see, there's a couple questions here. One was that from Cyrus, if the SVM SAG is more accurate than humans to detect the need for epilepsy surgery, why isn't it used clinically? So he's ready to jump ahead.
So that's a good question. And that entails validation. So I think we need to test that a little bit
And
more.
it's DAG, not SAG. Yeah.
Yeah. It's spoiler alert. Vince, Anis, myself, we applied for NIH funding for this. We got a good score. We're submitting it back again. And that's exactly to address the question you just mentioned. If we can test this, maybe that was willy-nilly findings with 90 patients, right? We don't know. We got to test this in a little bit more systematic way so that there is validation that this is not prone to error, which obviously, as you can imagine, could be potentially dangerous, right? So that's the idea. I should also mention that we don't support vector machine might not be as good as convolutional neural network. It's better than chance, but I don't think it's near as good as convolutional neural networks. Hence the need for this research.
Mm-hmm. A question from Adam, which is sort of the other side, is in what scenarios might AI fail in guiding clinical treatment?
That's a good question. So one thing that we don't know the answer to yet is whether some of the patterns that we see are related to having just a chronic neurological illness, right? So epilepsy is often associated with developmental abnormalities or sometimes neuropsychological problems such as memory problems, attention problems, etc. Chronic illness and several things associated with those. if that's what we're picking up, you could potentially think that it might not be specific to a disease and therefore not be accurate enough, and then say to the clinician like, oh, this person probably has temporal lobe epilepsy, when in reality he or she may have features associated with non-specific neurological injury. So again, in the spoiler alert category, one of the things that we're wanting to do that we haven't done yet, other than that Alzheimer's disease project that I mentioned before, is to carefully compare temporal lobe epilepsy with other forms of epilepsy, and then compare that same model with autism, with Alzheimer's disease. As you can see, this multi-step kind of cleaning out the other features is probably something that's very important.
Great. Other questions? And maybe Leo, you can unshare your screen as well so that we can
Excellent.
see larger views of everyone. Yeah, perfect.
I have a question. It's not about structure. It's more about activity and connectivity. So seizure basically changes brain activity, right? And I'm guessing I don't have any experience on the epilepsy. So these changes in brain activity is not just during the seizure. I'm just wondering how much study has been done on the effect of seizure and epilepsy in the brain activity and connectivity, and how much this type of AI approach can be used for clinical purposes.
Yeah, it's a great question. I mean, so that's that other project that I talked about at the very end is about that. So there are a few things that we know with epilepsy, and there are many things that we don't, of course. One of the things that we do know is that um, for somebody to have seizures, they need to have a hyperexcitable pattern within a network. And a network, it obviously means brain areas connected, right? Um, for some individuals, seizures can be very focal. In other words, they can just be associated with one region, but for many individuals, it stays focused and it spreads out. In in that context, I think it's important to think that not forms of brain plasticity are good, right? So brain plasticity, in the context of if parts of the brain wire together, fire together, wire together, right? If you have parts of the brain that are working together, they are more strongly emphasized and they remain more strongly connected. We think that happens with epilepsy in the context of bad neuroplasticity. So areas that are commonly associated with their reentrant pattern of hyperexcitability, they end up getting more and more and more and more and more connected. So there is a lot of evidence nowadays, and this is growing and picking up more momentum as well, is that individuals with epilepsy, even if seizures start in one area, with time, that propagation pattern within a given network makes that network stronger to the detriment at other brain regions. However, that network is not exactly the same to every single individual. It can be slightly variable, even though the epicenter might be similar, there can be a little bit of variation. The size, the location, the extent to which it spreads, obviously within specific anatomical regions, right? So insula, lateral temporal, medial temporal, etc. In the context of temporal lobe epilepsy, probably dictates some of the phenotypes. We haven't even thought about, at least I haven't, in terms of using, we thought about, but we haven't even started yet in terms of using, for example, connectome imaging in the context of convolutional neural networks. But that's certainly the next step, right? Because you can always use not only T1 weighted images, but you can also use their connectivity patterns. And then of course, there's a lot that could be done in this context, right? As well.
Got a question from Dong about would it be helpful to include additional modalities such as EEG or even SNPs?
Yeah, that's a great question. And I take that SNPs are genetic markers. Is that
Yeah.
Yeah. Yeah, I think so. And I think there are layers, right? So obviously the most immediate thing that comes to mind is multiple modalities in the context of imaging. So, as Armin was mentioning, using connectome imaging or things that the T1 weighted scan does not provide full information on. That's certainly the case. But most certainly, if we can also have layers related to additional features that are beyond imaging, I think that's definitely where this needs to go. I didn't have a chance to show this, but here, because it's a slightly side project, but we and other people have worked on how much can you predict if you use just clinical data, right? If you're ready to extract clinical data from a chart. And you can predict some, for example, in terms of surgical outcomes, but imaging certainly adds a lot to that. And much, if you could just predict based on charts, there wouldn't be a need for imaging and things like that. And we know that that's insufficient. But EEG, for example, it's another important part. So again, spoiler alert number 3 today. We're planning to do in this grant that we're resubmitting to evaluate the relationship between some of the neuroimaging features and some information that one can get from EEG, particularly intracranial EEG, particularly things that come from the brain itself. And that's more to validate the model in terms of, is this where the seizures are? If you see a salient pattern here, the deep learning model is picking up, is that really where the seizures are traveling, as opposed to using the EEG for classification, which certainly would be another step. But that's a good point. We just haven't done it ourselves.
Did you collect, say, resting fMRI data from these folks as well?
No, so that's a good question. Yes and no, Vince. We don't have resting state fMRI from a very large number of individuals. So I have from probably in the low hundreds, and that's from the research studies where Enigma, for example, doesn't have resting state. We have more DTI than we have resting state. But, again, there's close to, and I'm making a guess here, I need to check, but there are low hundreds numbers of those individuals. So it's not like in the thousands like there would be in Enigma, but not for everybody, because it's not commonly done clinically, I guess that's the point.
Right, yeah. There seems to be a lot of interest in multimodal features and covering patterns across structural data as well. I wonder if we'll do one more question. I guess from Mario, have you considered training a model with features extracted from different modalities? So different MRI sequences or EEG measurements. So having multiple markers. I mean, it seemed to me like in the results you were showing, you were particularly focused on spatial relationships, right? Laterality and location and things like that you can kind of get from the structural MRI, but the temporal information maybe could bump up the predictive accuracy even more. It's obviously it's more burdensome to have put all that together and collect it. But what are your thoughts?
Yeah, that's a good point. Obviously the resolution in terms of temporal components is with structural imaging is just not there, right? On the other hand, the spatial resolution from EEG is also suboptimal, particularly if you're looking at scalp EEG, or if you're looking at intracranial EEG, you don't have a lot of sampling across the entire region. So there is probably a lot that can be gained from it, because again, the domains are fairly different. In the context of epilepsy, or in the context of this particular form of epilepsy with EEG, one can engage about the location of seizure onset, broadly stated, and where it spreads to. And of course, using an orchestra analogy, you can see where the trouble in the music starting where it's moving to, and you can see, there's trouble in the music, whereas the picture of the brain is really a static picture of the orchestra, right? So I think there's a lot that could be done in that, but I would gauge probably more in the context of prognostication as opposed to diagnosis. That would be my guess.
Great. So thank you very much. We're right at 2, so we'll probably close the session for now. But as I think Armin had mentioned, and folks know, we'll have this the recording up, and there's also going to be this Slack channel where we're going to take the questions that people asked and put them there, and maybe there'll be some more, and we'll help you kind of sort of figure out how to connect into that. But thank you again for your time. This is a really interesting session, and thanks everyone else for joining as well. And enjoy your weekends. All right.